Reyes, and I'm going to present you the work Bioinformatic Strategy to build a transcriptome reference for PageSpam using short and long reads. I work at AgroSavia, la Corporación Colombiana de Investigación Agropecuaria. PageSpam, Pactis cassipaes code, is an orphan crop of American origin distributed in Central and South America. Here you can see the distribution of the wild and the land races. This palm has an estimated genome size of 4 gigabases and currently lacks a genomic reference. In this study, we use Illumina and Oxford nanopore sequencing to build a hybrid transcriptome for this crop. Here you can see the palms, and in this figure you can see the fruits of the peach palm. It has an important economy in the Pacific region of Colombia and other Central and South American countries. Here's the methodology. The workflow for constructing the reference assembly will first short and long reads running to multiple de novo assemblers. Here we present the best option for each case. For the nanopore reads, we also have an error correction step. Alongside, a reference-based approach using light kinesis annotation was used and integrated both short and long reads. And then, in parallel, a de novo complementary approach was proposed. This approach complemented used Illumina short reads and the best nanopore assembly. We aligned the Illumina reads to the best nanopore assembly, and the on-map reads were assembled using Trinity. Then we used Rattle for clustering this on-map reads assembly with the best nanopore assembly, and we obtained for assembly that we named Rattle plus on-map transcriptome. We compared these 4 transcriptomes using the Busco completeness score, RNA-QAS metrics, and the percentage of functional annotation. The results. Well, first, there's a difference between the amount of data. Long reads were the 4% of the data, while short reads were the 96% of the data. This is because we have traditional work with short reads, and this was our first attempt to work with long reads for transcriptomes. Here are the results of the assembly. In this bar plot, we have the completeness, Busco completeness score for the Liliopsida lineage, and we can see that the first is the Illumina and Trinity assembly. It obtained a 64.3%. The nanopore assembly obtained a 57.8%, and this difference is associated to the differences in input data that can affect de novo assembly metrics, promoting a complementary approach. The reference-based approach obtained a 63.4%, and then the combined approach obtained a 78.9% percentage. The 2 best assemblies, the Illumina and the Rattle plus on-map, had different fragmented scores. The first one, the Illumina, had a 23.2% fragmented scores, while the fourth assembly, the combined approach, obtained a 11.4. Then in this table, we present RNA-QAS metrics. And you can see that the nanopore assemblies have shorter minimum sizes. However, most of the transcript exceed the 500 bases. And so we selected the 4 assembly, the Rattle plus on-map, because it has the highest Busco completeness score with the lowest fragmentation. It had also a 73% of transcript that had a functional annotation. So for the conclusion, this study presents the first reference transcriptome for Bactis cassipaes cool, constructed from both short and long reads. And well, we think that additional long read data can enhance gene content in the transcriptome significantly without increasing fragmentation. We are trying to obtain more data. Thank you very much for your attention.